Assalamualaikum and hi everyone. So now we move to subtopic 6.5, alternative mechanisms of carbon fixation. So at the end of the lesson, students should be able to explain photorespiration and state the alternative mechanisms of carbon fixation, including C4 and CAM pathways, and compare carbon fixation in C4 and CAM plants. Photorespiration is a metabolic pathway that consumes oxygen and ATP, and it will release carbon dioxide and decrease photosynthesis output. One of the major problems facing terrestrial plants is dehydration. On hot and dry days, plants close the stomata to conserve water, but this causes problems for photosynthesis. On hot, dry days, C3 plants close their stomata to conserve water. Okay, so imagine here when the stomata is closed, so photosynthesis rapidly uses up the carbon dioxide remaining, okay, in the leaf remaining, and it will produce the oxygen. Okay, so when the stomata close, oxygen level, carbon dioxide level drop. Okay, carbon dioxide level drop as carbon dioxide is consumed in the Calvin cycle, and oxygen level rise as the light reaction convert light to chemical energy, okay, during photolysis of water. So in most plant, C3 plant, initial fixation of carbon dioxide, kalau normally okay, the fixation of carbon dioxide occurs via RUBP carboxylate oxygenase, and it will result in a 3-carbon compound 3-phosphoglycerate. Okay, so the plant example of the C3 plant is rice, wheat, and soybean. While for the RUBP, carboxylate oxygenase, when the oxygen ratio increase, okay, normally RUBP carboxylate oxygenase accept carbon dioxide, but when the oxygen ratio increase, it will bind the oxygen. Okay, RUBP carboxylate oxygenase akan add oxygen to RUBP. Okay, so oxygen here, okay, they act as a competitive inhibitor to carbon dioxide. Okay, so that's why Rubisco, they act not only as carboxylase but also as an oxygenase, because high level of oxygen, okay, high level of oxygen will compete with carbon dioxide for the active site of Rubisco. Okay, so during while photorespiration, RUBP carboxylate oxygenase add oxygen, okay, add oxygen to RUBP. Okay, so RUBP will split into two, one molecule of 3-carbon compound. Okay, the 3-carbon compound iaitu 3-phosphoglycerate, and another one molecule is the 2-carbon compound, okay, 2-phosphoglycolate. Okay. Okay, so then the two carbon okay, fragment ni akan exported from the chloroplast and degraded to the carbon dioxide. Okay, so this process, when you look, it occurs in the presence of light, and it requires oxygen like aerobic respiration. Okay, and it will produce carbon dioxide and water. So photorespiration does not produce ATP, so it reducing the efficiency of photosynthesis. It reduces photosynthetic efficiency because it removes some of intermediates used in the Calvin cycle. Okay, one molecule of 3-phosphoglycerate is produced instead of 2 molecule of 3-phosphoglycerate. Okay, sepatutnya dalam keadaan normal situation, okay, proses the fixation, proses fotosintesis ini akan hasilkan dalam Calvin cycle, dia akan hasilkan the 2 molecule of 3-phosphoglycerate, tapi bila berlaku photorespiration, dia akan hasilkan hanya 1 molekul 3-phosphoglycerate. Okay, hanya satu saja molekul trifosfoglycerate. Okay. Certain plant species have evolved alternate modes of carbon fixation to minimize photorespiration. In C3 photosynthesis, RUBP is carboxylated to form a 3-carbon compound via the activity of RUBP carboxylase oxygenase. So, for C4 photosynthesis, okay, other plants use C4 photosynthesis in which phosphoenol pyruvate or PEP, okay, is carboxylated to form a 4-carbon compound using the enzyme PEP carboxylase. It is named for the 4-carbon molecule present in the first product of carbon fixation in these plants, in contrast to the 3-carbon molecule products in C3 plant. So this C4 plant have crans anatomy leaf. Okay, the special character for the C plant, okay, they have the crans anatomy leaf. Okay, and the plant example for the plant is a corn and sugarcane. Okay, di mana it consists of the crans anatomy leaf for the C4 plant ni consists of mesophyll cell which is arranged concentrically around bundle sheet cell which also has chloroplast. Okay, so here is the structure of the mesophyll cell that located surrounding the bundle sheet cell, and it is consists chloroplast. It has a chloroplast. Okay, for this C4 plant C4 plant leaf. Okay, so kalau you compare dengan C3 plant, C3 plant does not have a crans anatomy leaf. So the plants that adapted to warmer environments have evolved two principal ways that use the C4 pathway to deal with this problem. So it will conduct C4 photosynthesis in the mesophyll cell, okay, and then the Calvin cycle, okay, in the bundle sheet of cell. So C4 pathway, also known as the H-SLAG pathway in C4 plants, involve the carbon dioxide twice. Okay, carbon dioxide is fixed twice. First, carbon dioxide fixation which occur in the mesophyll cell, and then the second carbon dioxide fixation which occur in the bundle sheet cell. So we look to the first carbon dioxide fixation. So this process occurs in mesophyll cell, which is the carbon dioxide will bind to phosphoenol pyruvate, okay, to form the oxaloacetate. So this process catalyzed by the PEP carboxylase, because so PEP carboxylase has a higher affinity towards to the carbon dioxide, okay, rather than the RUBP carboxylase oxygenase. Okay, so in this process, okay, actually why this plant called as C4 plant? So this is because the first product of carbon fixation, okay, the first product of carbon fixation in this process is oxaloacetate. Okay, ni oxaloacetate ini adalah the 4 carbon molecule. So that's why this plant is called as C4 plant. Okay, and C4 plant will run the C4 pathway, okay, mechanism. Okay, and then after the fixation of PEP carboxylase, when the PEP carboxylase catalyze the process fixation of the carbon dioxide, to the phosphoenol pyruvate. So the OAA that produced will reduce, okay, will reduce to malate. Okay, OAA is reduced to malate. Then this malate is transported to the bundle sheet cell. In the bundle sheet cell, the second carbon dioxide fixation occurs. Okay, malate undergoes decarboxylation. Okay, malate undergo decarboxylation to produce pyruvate. So here malate undergo decarboxylation to produce pyruvate and carbon dioxide. Okay, so then pyruvate will return to the mesophyll cell where it is phosphorylated by ATP. Okay, phosphorylated by ATP. So ATP transfer it phosphate to pyruvate to form phosphoenol pyruvate, thus completing the cycle. While for the carbon dioxide, the ability carbon dioxide will combine with the RUBP and undergo Calvin cycle. Okay, so the ability of phosphoenol pyruvate carboxylase, okay, to bind with carbon dioxide. Okay, here the PEP carboxylase that can bind carbon dioxide even the concentration of carbon dioxide is very low. So this process can increase the carbon dioxide in the bundle sheet cell. So carbon dioxide here will increase in the bundle sheet cell. Okay, thus enable Rubisco or RUBP carboxylase, okay, in the Calvin cycle to fix carbon dioxide instead of oxygen, okay, with the RUBP and enters the Calvin cycle normally. So the tendency of photorespiration here can be reduced. Okay, so next, seterusnya for the crystallization acid metabolism CAM. So this is an adaptation in hot and dry condition, also evolved in succulent plant. Okay, these plants open their stomata during the night and close stomata during the day to conserve water and prevent entry of carbon dioxide. So, but open at night to collect the carbon dioxide. So this mode of carbon fixation is called crystallization acid metabolism (CAM), and it occurs in mesophyll cell. Okay, so example the cactus and pineapples. Okay, so during the night, okay, CAM plants open the stomata. Okay, so it will open the stomata. Carbon dioxide will enter and fix to the PEP. Okay, combine with the PEP to form the oxaloacetate, okay, OAA. And this process is catalyzed by PEP carboxylase. OAA, okay, so the OAA, so OAA then reduced to malate or malic acid, okay, malate, and the malate are stored in vacuoles until morning. Okay, so dia akan OAA then reduce to the malate, then malate will transported, okay, ataupun disimpan dalam vacuoles until morning. Okay, so during the day, okay, CAM will close the stomata. Okay, so what happened? The malate from the vacuole, okay, malate or malic acid dalam vacuole ni, dia akan release, okay, malate akan release from the vacuole and decarboxylated. Okay, dia masuk ke dalam chloroplast semula dan berlaku decarboxylation lah, okay, releasing of the carbon dioxide. Okay, malate form the carbon dioxide and the 3 carbon compound. Okay, carbon dioxide is released to the Calvin cycle, masuk dalam Calvin cycle, and akan run proses Calvin cycle tu seperti biasalah. Okay, the light dependent reaction supply ATP and ADPH to the Calvin cycle. Okay, so dia boleh undergo proses Calvin cycle tu seperti biasa, tak ada masalah. So kat sini cara dia untuk mengelakkan berlakunya proses photorespiration untuk tumbuhan jenis succulent lah, macam pokok kaktus. Okay, so dia boleh juga jalankan proses Calvin cycle, tapi dia kena lalu CAM pathway. So this yang berlaku pada waktu malam, kemudian malik asid disimpan dalam vakuum sebelum tunggu waktu siang. Itu adalah waktu siang dia akan riliskan malik asid ni, dia akan riliskan carbon dioxide lah. Malik release from vacuum and decarboxylated. Dia riliskan carbon dioxide, dan carbon dioxide ni barulah rilis masuk ke dalam Calvin cycle. So ini adalah CAM pathway lah untuk tumbuhan jenis contoh macam pokok kaktus.